Professor Paulson, for the introduction, and thank you all for coming out today. I know you have your final presentations tomorrow, so I think it's a very good turnout for that. So as Dr. Paulson mentioned, I'll be talking more about how we can kind of extend the COBRA framework to account now for macromolecule resource allocation. So one of the problems that we deal with as bioengineers is to develop bioprocesses in order to make a chemical or a protein of value. And to do this, we want to focus usually on at least 3 or maybe 4 different bioprocess objectives, which is yield, how expensive is it to actually make this product from the feedstock, the rate, how fast, how much you can make it, the titer, which is the total concentration volume of how much of the product you can make, and the actual product's quality. You need to make sure, especially for macromolecules like therapeutic proteins, you need to make sure that the protein does what it's meant to do. And so the bioprocess objectives of yield, titer, and rate really depend on now the cell factory objectives of yield, rate, and of course the quality of the product. And so we can address the problem of bioprocess objectives with now the cell factory objectives, and to actually try to design a cell factory, an engineered microbial strain or a mammalian cell line. We can try to address each of these objectives one by one. So we can first consider maximizing the yield. So let's say we want to make 1 propanol from glucose, and we wanted to maximize the yield of this product from its feedstock. How would we go about doing this? Well, we know from taking Bioengineering that we could take our host cell organism, in this case it's E. coli, and reconstruct a metabolic network based on its annotated genome, and then include not only the native host metabolic network, but also the heterologous pathway reactions within the reconstruction. And then we can try to maximize the yield. And to do so, we can convert the metabolic reconstruction, the network, into an optimization problem. In this case, it'll be a simple linear program with the constraint matrices expressed as the stoichiometric matrix there. And we would try to now, and of course there are, as you know, many resources to actually do this step. And then we would try to solve the optimization problem to maximize the yield. And so what we get in this case, in this particular example, is we find out even though there are 2 possible pathways to make propanol from glucose, there's one native pathway from threonine, and there's a heterologous pathway from citramalate. It turns out that the way to maximize the yield is to actually use both pathways simultaneously. And it turns out you get at least 33% greater theoretical yield if you use both pathways simultaneously than if you use just one of these pathways individually. Okay, so maximizing yield, pretty simple problem. The next question we want to ask is, what if we wanted to maximize the overall rate of production, which is actually a distinct problem from yield. Because you can have an extremely high theoretical yield, but your rate of catalysis might be really, really slow. That really won't do for a bioprocess. So the rate of any given reaction, of course, depends on the catalytic efficiency of the enzyme, so the turnover. So it's going to be kcat over km. We can kind of combine all the different factors into an effective rate constant. And what we end up with is essentially, in addition to our base formalism of SV equal to 0, the mass balance on all of the metabolites, and the bounds on each individual reaction flux, we can also impose a constraint that is limiting the maximum rate of each reaction by its catalytic efficiency. And so by effective rate constant, we usually mean if we have, for example, a second order reaction, if one of these substrates is in excess, we would apparently have a first order reaction in the concentration of the substrate that is not in excess. In our case, it's actually more, you can think of it as the substrate being, the enzyme is saturated. And so the rate of the reaction will really depend proportionally on the amount of enzyme that you have. So that's what we mean by the effective rate constant here. And we apply this constraint for essentially all of the proteins, all of the enzymes in our reconstruction. And so we have this flux is limited by the catalytic efficiency for all of the enzymes in our proteome. And so that's what we have here. Now, when we do this, we also realize, though, that the amount, so now we have two different variables, actually. We have the flux of the reaction V, which you're used to, and we now have the enzyme concentration, which is now another variable. And you're going to have, each of these enzyme abundances for every protein in the entire proteome. And the point here being that we know that the total proteome mass of any cell is finite. And so we need to now constrain the total mass of the proteome by the observed total mass of the proteome, which is going to be P here, and M, that's just the molecular weight of each enzyme. And so what this gives us is essentially a total proteome constraint, which is essentially related to a macromolecular crowding. So the cytosol has a limited volume, and each enzyme has a molar density, and therefore you can only pack so many enzymes within this limited volume, cytosolic volume. And something very similar applies to the membrane, but in that case you'd be considering the surface area instead. So overall we have these proteome capacity constraints. And what we can do now is we can extend our metabolic reconstruction with now these macromolecular capacity constraints. The first one being, of course, the flux capacity, which is a function of the turnover, and the proteome capacity, which is a function of the molecular weight of each enzyme. And you can start to see that if you want to make more of one of these enzymes, for example, an enzyme within your heterologous pathway, because you want to speed up the rate of that product synthesis, you're going to have to increase the enzyme abundance for that enzyme, but because the total proteome is capped, you're going to have to necessarily decrease the abundance of some other enzyme. And so now what we end up with is a resource allocation problem in the context of these macromolecules, these proteins. And so this is the general framework of proteome capacity, proteome-constrained FBA. And there are specific implementations of this general framework, such as I mentioned briefly, molecular crowding, where we actually convert these masses into volumes, and also moment, which actually takes this approach directly. So when we impose these additional constraints, we're necessarily going to shrink the solution space, and in fact we're going to refine it, and usually our predictions are going to become more accurate. So if we have, so on the left what you're seeing is that the substrate is essentially, the enzyme is saturated, so the flux is now proportional to the enzyme concentration. And we start to see some more complex phenotypes where the growth rate is not just linearly proportional to the substrate uptake. Without these constraints, what you probably have to do in your assignments is you have to limit the lower bound or the total uptake on, say, your limiting carbon source like glucose. You have to set it to some arbitrary value of -10 or -20 for acetate. Those values actually come from experimental data. But if you were to provide double the glucose, you would see essentially doubling of the growth rate. But if we impose these proteome capacity constraints, we don't have to limit the glucose uptake rate anymore. It'll actually, if you get all of your rate constants correct and you get your total proteome mass correct, you'll actually be able to predict de novo the uptake rate more or less correctly. So that's the first advantage of adding these constraints. And we can go on to predict even more complex metabolic behavior. So if we were to grow E. coli in a complex medium with 5 different carbon sources, glucose up to glycerol, and if we didn't have these capacity constraints, what you would usually see is the model would try to take up all the carbon sources at once, as much as it can, and will try to maximize its growth rate. But we just saw that the total proteome is constrained, and if you wanted to express the pathways, the catabolic pathways for utilizing this one carbon source, you would have less proteome available to utilize to express the pathways needed to catabolize another carbon source. And so the optimization problem, which used to just be SV equal to 0, now the optimization problem becomes a bit more interesting because it needs to choose which enzymes to express that will actually maximize its growth rate at the cost of not expressing other enzymes, which means it chooses not to utilize certain carbon sources. And then if you do this in a dynamic way, where you start with all carbon sources available, choose the best carbon source, or maybe sometimes a combination of different carbon sources to maximize your growth rate, use those carbon sources up, you've depleted those, move on to the next step, and then you solve the next optimization problem of choosing what the best combination of carbon sources are. And so on the left panel is actually the experimental measurements, and on the right panel are the simulations that were done using the FBA with molecular crowding that I mentioned a couple slides ago. And you can see that not only are the qualitative trends, pretty good, but the actual order by which the model predicts which carbon source to use first, and even combinations of carbon sources, as you can see here. So glucose is the best one, so it'll just use glucose. Also interestingly, it's already doing acetate overflow metabolism here. And then it uses 2 different carbon sources, which the model thinks are the next best 2 to use at the same time. And then it'll start using glycerol and, actually galactose, and then finally glycerol. Once it uses up nearly all of the primary carbon sources, it'll actually start reutilizing the acetate that is secreted before. So this is a really interesting, pretty complex phenomenon. And just by adding those 2 lines of constraints, you can more or less reproduce this behavior. All right. So this proteome constraint and the flux capacity constraint is more or less ubiquitous. It doesn't just apply to E. coli. It's also it also applies for mammalian cells and also for even human cells. And there's a nice set of references if you're interested, where you can learn more about different organisms where these constraints have been investigated. All right. So far we've talked about maximizing theoretical yield, and we've talked about maximizing the rate, and realized that because the total proteome is limited, we need to actually we need to solve a pretty sophisticated resource allocation problem on the entire proteome scale in order to maximize rate. But let's revisit the maximization of yield problem now, because now we know not only are the fluxes limited, but also the proteome. But so far we've only considered the protein concentrations as variables that aren't really tied back to metabolism. Essentially, we know that the proteins are actually created. They're synthesized through the metabolic network. The metabolic network provides the precursors for these enzymes, amino acids. And so what we can now do is to actually add on to our protein-constrained FBA the network of protein expression. So that's an extra layer of reconstruction. And I think you all know where this is headed. It's essentially the me models that Colton described a couple of weeks ago. So let's just go through this just step by step. So we started with, let's see, the metabolic network and enzymes. So we can add on essentially fluxes for translation. And we'll just do a mass balance on each protein. We'll say synthesis minus its consumption is 0. So there's no accumulation or depletion. So dilution is going to be essentially the sole source of consumption of a protein. There can also be protein degradation, but let's not consider that for now. And dilution is simply the growth rate multiplied by the concentration of the enzyme. So it's getting diluting it. So now we have growth rate times the enzyme concentration, and we have a new flux for translation. And what we can do next in terms of the translation flux, well, the translation flux is now another consumer of an amino acid, which also needs to be mass balanced within the entire metabolic network. And so we can add another mass balance, or we can expand the mass balance, the row for each amino acid, by adding in a column, which is the translation flux, and simply say synthesis minus however many amino acids you need to make this particular protein, and there, right there, minus all the metabolic processes that consume this amino acid, and you'll have a mass balance on the amino acids. All right. And so if you add in these two expanded mass balance constraints into what we just had before, now you have what's starting to look a lot like more of a full reconstruction of the expression matrix. Right now we've just added in translation, which is just going to use amino acids. And then, of course, we can now consider not just translation, but all of the different steps for expressing a protein. And we can expand this process furthermore. We can go in even more detail, look at all 27 or so processes or subsystems that comprise the entire expression network. And by the way, this process, of course, is essentially the same as how you would reconstruct a metabolic network, except because a lot of these processes are actually amenable to some kind of a template format. You basically give it the number of nucleotides or the number of amino acids to express this gene, this sequence. You can kind of semi-algorithmically generate the expression network. And so you end up with now kind of an integrated framework starting from just the metabolic reconstruction. And we've added in the flux capacity constraints. And then we've added in the total macromolecular capacity constraint. We just looked at the proteome so far, but as I mentioned, you can also look specifically at the surface area being limited within on the membrane. You can look at the periplasmic proteins, and we have our macromolecular capacity constraint, and then we can add on the entire reconstruction of the expression machinery. And what this allows us to do then is to address an extended set of hypotheses and predictions. And so, I've often been asked, why would you go through the additional effort, say, of expressing the entire expression network? We already saw we can get quite a bit of refinement in our predictions just by adding steps B and C. And now we're saying we also want to add in the finer grained reconstruction of the expression network. So the most obvious use case is if the product of interest is not a small molecule, but we actually want to make a protein, like a therapeutic protein. It now becomes quite feasible, right, to actually say, reconstruct my metabolic network, reconstruct my expression network, and add in the heterologous pathway that'll actually make this particular protein, which in this case is just going to be, here's the sequence of the protein I want to make, get my expression machinery reconstruction to make this sequence of protein. So it's really becomes trivial once you have your M and the E reconstructions to actually simulate protein synthesis in your model, which we couldn't do before if we just had a metabolic network. So that's that's easy to see. Also, if you suspect that, you see a pretty interesting phenotype, and you suspect that the mechanism underlying this phenotype really lies somewhere in the expression network, there might be, certain proteins might not be operating at their proper capacity. There might be some folding that isn't really being performed properly, and so on. So then you would actually need to reconstruct the expression network to actually address those questions. And of course, if your conclusions, if you think they're really sensitive to how costly it is to make a certain protein, then you probably want to be able to compute rather precisely the expression cost, which is also going to incur a metabolic burden to actually make different kinds of proteins. Right. And of course, if we want to compare our predicted protein abundances or transcript abundances with omics data, proteomics and RNA-seq, for example, then having a full integrated reconstruction is actually going to help you there as well. All right. So now that we have this overall framework to account for macromolecule allocation, we can start to address some more difficult questions, like the quality of proteins that are being expressed. So one interesting example is what happens to proteins, to individual proteins and the proteome overall under different stress conditions. And right now we've looked at what happens when cells are thermally stressed, when they're exposed to very high temperatures. And what it turns out that in this case the growth of E. coli is actually pretty sensitive to the temperature. You may have known this already if you based on experimental data. But if we look at this plot here, we have temperatures ranging from 24 up to 46 degrees Celsius, and this is the relative growth rate relative to, let's see, I guess the optimal at 37. Okay, so there's quite a bit of growth rate dependency on temperature, all the way down, decreasing by more than 70%. And so, something's going on when cells are thermally stressed. Well, if we wanted to reconstruct and actually simulate this temperature-dependent behavior of E. coli, we would first need to figure out, well, what does temperature affect within the cell? It turns out that when proteins are thermally perturbed, several of their characteristics are actually affected. And those characteristics include their propensity to aggregate together, and they won't be functional anymore. Their tendency to fold or misfold, and also their catalytic efficiency. So all three of these properties of proteins are actually going to be affected by temperature. And furthermore, there's actually a pretty elaborate maintenance network within E. coli that is dedicated to ensuring that proteins are in fact properly folded, that they're not aggregated. And so this chaperone network is now an additional factor we need to consider. So what we can do is we can now actually reconstruct this chaperone network, or this protein's homeostasis or proteostasis network in E. coli. And so that's what we have here. And we can furthermore make these different properties of the proteins a function of temperature. So the simplest one to look at is perhaps the catalytic efficiency being a function of temperature. So that'll be essentially the K effective rate constant is some function of temperature, and we have a thermodynamic model to actually compute this, to actually formulate this function. So once we've figured out the mechanisms by which temperature affects E. coli proteins, we can then go ahead with the reconstruction and actually add the constraints like we've done before in the first half of this talk, add the constraints in to make the rate constants a function of the temperature. And once we've done all of that, we can now start to predict temperature-dependent behavior of E. coli metabolism and growth rate. And so what we see here is that if we perturb the temperature, we actually see now completely different pathway utilization, metabolic pathway utilization. The low temperature and in the really high temperature. And what's notable here is that under high temperature, for example, thymine or thymidine cannot actually be utilized anymore. So the cell can't really take up thymidine because the pathways that are needed to utilize it are now inactivated, essentially. And so we actually have experimental data to support this prediction, where under low temperature you can actually use thymidine and glucose, of course. And under high temperature, you can't use thymidine anymore. Okay. So we can also now look at not only the metabolic pathway utilization, but also how the entire protein is reallocated under these two different temperatures. And so we go from a really small fraction of chaperones being expressed and used at the optimal temperature, 37, and a much, much higher requirement for protein maintenance through these chaperones at the higher temperature. And of course, this means that there's going to be more or less a trade-off between the proteins that would have been used optimally under the 37 degrees condition, and those proteins are now not really being used anymore under the highly thermally stressed condition. So this model, of course, is pretty complex, as you've seen, not only in terms of the reconstruction, but also in terms of the additional equations and the different thermodynamic models that go into it. But one thing that's interesting about these complex reconstructions is that you can use them in a way like a model system, where you can now perform a lot of different simulations under a lot of different perturbations to kind of distill the behavior of the cell, in this case as a function of temperature, into a relatively simple set of descriptions. In this case, we can say there's a simple growth law that relates temperature to the fraction of the proteome that is allocated to ribosomes, to chaperones, and to metabolic enzymes. And we have, in fact, a very simple linear relationship that actually connects all three of these macromolecular resources. So just to summarize, today we've kind of gone through step by step, starting from a metabolic reconstruction. And then we considered how individual enzymes are actually limited in their maximum flux due to a catalytic efficiency. And then we looked at the total macromolecular crowding and the proteome constraint. And then we looked at how proteins can be maintained and how we can actually reconstruct expression networks and maintenance networks to actually simulate the cell behavior with those systems in place. And we've also furthermore discussed how we can, once we've identified each of these biophysical constraints, we can express them as mathematical constraints within this within the COBRA framework. once we have this extended framework, extended model, we can use it to actually address, cell factory objectives, bioprocess objectives, even pretty complex ones like how to maintain the quality of your product under different stress conditions. And we were able to actually reproduce pretty complicated metabolic behavior phenotypes and even different stress responses. So my intended learning objectives for this lecture was really for you to be able to walk out and look at your problem of interest and think, is there a macromolecular basis for the phenotype that I'm observing? Is there a reason to consider a flux capacity constraint? Is there a reason to consider a limited amount of proteome that is finite within the cell? And then once you've identified this constraint, to actually go in and formulate, extend the COBRA methodologies that you've already learned within this course, and extend it. As you've seen, mathematically, it's pretty not too complicated. It's not very complicated. And so my hope is that you were able, walking out, you'll be able to actually identify the constraints and be able to formulate them for your own problem of interest. And with that, I'll be happy to take any questions. Thank you.